In this video, we will cover a general overview about the glycan changes in the tumor. So, in some of our previous videos, we have discussed how this aberrant glycosylation occurs in the case of pancreatic cells, especially in the case of this during metastasis. So, let us start with how the glycans looks like in the case of normal cells. So, these are the N glycans that are mentioned here, and this one is the complex glycans and with this fucose attached, and this one is the O glycans, and this O glycans in details as mentioned on this mucin glycoprotein that we can see here on the top of this glycan structures, and these are also the N glycans that is high mannose N glycans, and we can see that in the case of this mucin the glycans are silated, and in the normal case there is a presence of st3-gal1, and that helps in the addition of this galactose, and then this silation will occur afterwards. So these are the glycans that are usually found in the case of normal cells, and in the case of pancreatic cells what happens is that, let us start with this complex glycans. In this case there is a high branched fucosylated branched and glycan is usually observed in this. In the case of normal cells we can see there are only two branch, whereas the example here in the case of cancer cells shows that there are four branches. Along with this change in the fucosylation branching from the second glycan monosaccharide to the first glycan monosaccharide that we can see here. And also, since in the case of cancer cells we have seen from our previous reviews that there could be dysregulation in the C1-GALT1 gene, and that prevents this addition of galactose and the further formation of and the further O-glycosylation. And this can be seen here that the galactose is absent, and it will directly leads to the formation of this kind of structures, directly addition of sialic acid to this GalNAc structure. And also with the absence of C1-GALT1, this only GalNAc structures are also found, and this galactose and in some cases these GalNAc and galactose structures are found, and these are known as the T antigen carbohydrate. And the absence of C1-GALT1 can be seen with the absence or dysregulation of C1-GALT1 can be seen with this upregulation of this STN structure, and also the only presence of this GalNAc carbohydrates which are known as TN structure. And these are all usually found in the cancerous cells. And few more unique antigens that are found in the cancerous cells is, one of them is this S. levis A structure. In this case, this O glycans, there is a fucosylation has been observed in the case of O glycans that can be seen here, that is presented here in the O glycosylation of the mucin glycoprotein. And also to this complementary to this SLE A structure, there is also a SLE X structure that is not shown in this figure. That is and that structure is also found in abundance in the case of these cancerous cells, whereas these structures are absent in the case of normal cells, and these aberrant or abnormal structures are highlighted by this blue color, such as this TN antigen. We cannot see the TN antigen in the case of normal cells, and the STN antigen, and both of them could be related to the dysregulation of C1-GALT1. And we can see because of the other biological changes, we can see some other glycosylation, some other unique glycosylation, such as this SLEA glycosylation, and also because of the dysregulation in this end-linked glycosylation pathway, we can also see these heavily branched fucosylated and glycans. So I hope this video might have provided you the overview of what we are discussing in the past videos about this aberrant glycosylation in the tumors, and some of the aberrant glycosylation that I have already mentioned are highlighted in this light blue color. If you need to understand more about these glycan changes in the case of tumor, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.